video reviews priming the BVS 5000 blood pump. If you are unfamiliar with the BVS 5000, please review the system overview video, which is part of this training series. The BVS 5000 blood pump is packaged in a tray with a sealed Tyvek lid. This lid is peeled back to expose the sterile blood pump. The pump is held in place by a plastic retainer, which must be removed to access the pump. The tapes marked with lift labels are peeled back, and then the retainer is removed. The blood pump is then lifted out of the tray and placed in the sterile field. Beaded wraps secure the tubing on the blood pump. These wraps are removed as soon as the blood pump is in the sterile field. The blood pump is packaged with latex tubing and 1/2 to 3/8 inch downsizing connectors. These materials facilitate attachment to the priming circuit and are discarded after priming is complete. Arrows on the blood connectors indicate the direction of flow. Blood flows from the patient into the top of the pump and then into the atrial bladder. Blood passes through the inflow valve during pump diastole and into the ventricular bladder. The ventricular bladder ejects blood through the outflow valve and back to the patient during pump systole. The priming volume of this circuit is 600 milliliters. BVS 5000 blood pump is primed by circulating fluid through a cardiotomy reservoir. The perfusionist must supply 2 sterile 4-foot lengths of 3/8 inch tubing, 1 standard cardiotomy reservoir, and 2 liters of priming solution. Crystalloid solutions are most frequently used to prime the BVS 5000 blood pump. Albumin and packed red blood cells are sometimes added to the solution at the clinician's discretion. The priming circuit's components are placed in the sterile field and the necessary connections are made. 1 length of tubing is attached to the bottom of the cardiotomy reservoir and the inlet to the pump. And the remaining tubing is attached to the top of the cardiotomy reservoir and the outlet of the pump. The connections must remain in the sterile field at all times. A clamp is placed on tubing attached to the bottom of the reservoir. The reservoir is filled with approximately 2 liters of priming solution. Once the reservoir is full, the blood pump is inverted and raised to the level of the reservoir. The clamp released and the blood pump is slowly lowered. This permits the gradual displacement of air inside the blood pump and ultimately reduces the total time required to complete the procedure. As soon as the blood pump is full, it is returned to its proper position. The drive line is attached to the console and the pump control is turned on. The blood pump will now circulate the prime through the reservoir. The perfusionist should closely examine the valve sinus areas for trapped air. If air is present, briefly invert the pump while it is running. This will permit the air to float out of the sinus and into the reservoir. If bubbles are adherent to the inner surfaces of the pump, light tapping with a clamp will effectively dislodge them. Once the perfusionist is satisfied that all air has been removed, circulation of the prime should be continued for at least 5 additional minutes. The blood pump is disconnected from the priming circuit when the cannulae are primed and ready for connection. Circulation through the reservoir is stopped and the connections to the circuit are isolated using 4 tubing clamps. The latex tubing and the blood pump tubing on each connection is cross-clamped. The latex is then rolled off and the connector is ready for attachment to the appropriate cannula. The connections to the cardiotomy reservoir should remain in the sterile field until the entire procedure is completed. The same circuit may be reused should additional priming become necessary. A second blood pump can be rapidly primed by adding 600 milliliters of solution to the cardiotomy reservoir and attaching the pump to the existing circuit. Repriming a pump is also expedited by connecting to the existing circuit. During pump startup, the perfusionist should pay careful attention to the tubing leading into the blood pump. If any signs of air are visible, immediately clamp the atrial cannula or inlet tubing. This will stop flow through the pump. Turn the pump off and assess whether any air actually entered the pump. If the air is isolated to the atrial cannula and inlet tubing, the cannula and tubing can be disconnected and the bubbles removed by irrigating the junction. If air has entered the pump, then both cannula must be disconnected from the pump and the pump must be reattached to the priming circuit. The pump is turned on and the priming solution is recirculated. Successfully reprimed, the pump can be reconnected to the cannulae.